If you were to travel back through time, back beyond the age of dinosaurs, beyond the first forests, to a moment when the land itself was still learning what it meant to be green, you would find mosses already there, quietly pioneering a world that had known only stone and water. Fossils preserved in ancient rock tell us that mosses appeared around 450 million years ago, making them among the very oldest land plants we know. They were part of that extraordinary transition, that momentous leap when life moved from the safety of water onto the harsh and unforgiving surfaces of bare land. In those early days, the continents were empty places, swept by wind and baked by sun, and it was plants like mosses that began the slow work of making those places habitable. These humble plants belong to a division called Bryophyta, a group that also includes their close relatives the liverworts and hornworts, all of them sharing a similar architecture and approach to life. For a long time, scientists assumed that all mosses descended from a single ancestor that made that crucial journey from water to land, but genomic studies in recent years have suggested something far more intriguing. It appears that several moss lineages may have evolved on land independently, each solving the puzzle of terrestrial life in its own way, each arriving at similar solutions through the mysterious process of convergent evolution. This tells us that the move to land was not a singular miraculous event, but perhaps something more inevitable, something written into the possibilities of life itself. The innovations that allowed bryophytes to survive on land were profound. They evolved a waxy cuticle, a protective coating that wraps around their cells and slows the loss of precious water to the air. They developed protected reproductive structures that could withstand drying winds and intense sunlight. These adaptations, though they may seem simple compared to the elaborate systems of flowering plants, were revolutionary in their time. And there's something else, something hidden in the very code of their being. Whole genome duplications occurred in moss lineages, events where the entire genetic blueprint doubled, creating new possibilities, new combinations, new pathways for evolution to explore. These duplications help drive the remarkable diversity we see in mosses today. And that diversity is truly staggering. Among land plants, mosses occupy one of the most species-rich positions, second only to the flowering plants that would not appear on Earth for another few 100 million years. When we count the mosses that have been formally described and add estimates for those still waiting to be discovered and named, global moss diversity may reach around 20,000 species. 20,000 variations on a theme, 20,000 different ways of being small and green and persistent. These species occur on every continent, in nearly every terrestrial ecosystem you can imagine. They are absent mainly from the open ocean, for they remain fundamentally plants of land and fresh water, but wherever there is solid ground and at least occasional moisture, mosses have found a way. Even in Antarctica, that frozen continent at the bottom of the world, mosses dominate some ice-free coastal areas where few other plants can survive. They cling to rocks warmed by the brief polar summer, their dark cushions absorbing what little heat the sun provides, creating tiny oases of life in a landscape of ice and stone. It's a testament to their resilience, their ability to endure extremes that would kill most plants within hours. While flowering plants may dominate our attention with their colors and fragrances, their fruits and their partnerships with pollinators, mosses have quietly achieved their own empire of diversity, an empire built not on showiness but on subtle perfection of form and function. The exact number of moss species remains uncertain, as new forms continue to be discovered and described. Conservative estimates place the number around 12,000 known species, but when we account for the many forms not yet formally described, not yet studied and named by science, the global diversity may reach 20,000 species or more. Each of these represents a unique solution to the challenges of life on land, a particular way of being a moss that works in a specific context under specific conditions. Mosses have claimed territory on every continent, from the tropics to the poles, from sea level to high mountain peaks. They inhabit nearly every terrestrial ecosystem, absent mainly from the open ocean, which remains the domain of their algal ancestors. In rainforests, they drape from branches and coat fallen logs. In deserts, they hide in the shadows of rocks, appearing to vanish during drought and returning to life with rain. In temperate woodlands, they carpet the forest floor in green so deep it seems to hold shadows even in brightest daylight. Perhaps nowhere is their tenacity more evident than in Antarctica, where in certain ice-free coastal areas, mosses dominate the landscape. Here, where few other plants can gain a foothold, where temperatures regularly plunge far below freezing and the growing season is measured in brief weeks, mosses persist. They form communities of stunning resilience, their slow growth adding microscopic layers year after year, building cushions that may be decades or even centuries old. In these extreme environments, mosses don't just survive, they thrive, becoming the foundation of entire ecosystems, the primary producers that support whatever other life can exist in such challenging conditions. Understanding mosses requires us to set aside much of what we know about other plants. Mosses are non-vascular, meaning they lack the specialized tissues called xylem and phloem that other plants use to transport water and nutrients throughout their bodies. Trees and flowering plants rely on these internal plumbing systems to move resources from roots to leaves, to grow tall, to reach toward light. Mosses have no such luxury. They cannot build internal highways for water, cannot pump nutrients from one end to the other. Instead, they have evolved entirely different strategies for survival. Where other plants send roots deep into soil, mosses produce rhizoids, delicate hair-like structures that serve not to absorb water and nutrients, but primarily to anchor the plant to whatever surface it inhabits. These rhizoids grip rock and bark, concrete and soil, holding the moss in place against wind and rain, but they are not roots in any conventional sense. They lack the complexity, the specialization, the absorption capacity of true roots. Mosses take in water and nutrients not from below, but from above and all around, absorbing what they need directly through their leaves and stems. When you look at what appears to be a single moss plant, a unified green cushion or carpet, you are actually observing thousands, perhaps tens of thousands of individual plants living in intimate proximity. Each tiny shoot is a separate organism, but they live so closely together that they function almost as one, sharing water, creating a microclimate within their collective structure, protecting each other from extremes of temperature and desiccation. This colonial existence is key to moss survival, as isolated individuals would quickly succumb to stresses that the community can withstand. Despite their lack of vascular tissue, moss stems are not without structure. They contain specialized cells called steroids, which provide support and stiffness to these tiny shoots. Though each individual stem may be only millimeters tall, the steroids allow it to stand upright, to hold its leaves toward the light, to maintain its form against the weight of water droplets and the pressure of neighboring plants. It's a miniature architecture, but architecture nonetheless, precisely calibrated to the scale at which mosses operate. Some moss leaves are marvels of minimalism, constructed of a single layer of cells. These leaves, only one cell thick, allow for the direct absorption of water and nutrients from rain, fog, and dew. There is no barrier between the environment and the living tissue of the plant, no distance for resources to travel. What lands on the leaf surface is immediately available to the cell beneath, or rather, is immediately available to the cell itself, for in these single-cell thick leaves, surface and interior are one and the same. The dense arrangement of leaves and rhizoids in a moss colony creates countless tiny spaces, capillary gaps where water can be trapped and held through surface tension. When rain falls or fog condenses on a moss patch, this network of spaces acts like a sponge, capturing and retaining moisture long after the air has dried. This stored water becomes a reservoir from which the moss can draw during dry periods, extending its ability to remain active and photosynthetic between rain events. The architecture of the colony itself becomes a water management system. Living without protective bark or thick cuticles means that moss cells face environmental extremes directly. When temperatures drop, moss cells freeze. When the sun beats down, they heat. When drought comes, they desiccate. This direct exposure could be fatal, but mosses have evolved remarkable tolerance for these extremes, able to shut down their metabolism when conditions become hostile and resume normal function when favorable conditions return. Their cellular machinery can withstand freezing, drying, and heating that would kill the cells of most other plants. The forms that mosses take are wonderfully diverse. Some grow as tight cushions, rounded domes of green that shed water and resist compression. Others spread as carpets, flat expanses that cover ground or rock in continuous sheets. Still others hang as pendular strands from tree branches, swaying in the breeze like miniature curtains. Some species grow in tiny tree-like forms, with a distinct stem and radiating branches, perfect miniature forests in themselves. Each growth form represents an adaptation to particular conditions, a way of arranging leaves and stems to maximize survival and reproduction in a specific context. The shapes of individual moss leaves are equally varied and equally purposeful. Some leaves are long and narrow, others broad and rounded. Many have teeth or serrations along their edges. These features are not decorative but functional, controlling how water flows across the leaf surface, how quickly it evaporates, where it accumulates. The precise architecture of each leaf has been refined by evolution to manage the most precious resource in a moss's life, the water upon which everything depends. Among the most specialized mosses are those that live on trees, the epiphytes that spend their entire lives perched on bark, never touching soil. These tree-dwelling species have evolved exquisite precision in their habitat selection. Some prefer the rough bark of oak or maple, others the smooth bark of beech. Some colonize the tops of horizontal branches where light is plentiful, others the shaded undersides where moisture lingers. Some nestle into the forks where branches meet trunks, accumulating the organic debris that collects there. Each species has found its niche, its particular combination of light, moisture, and substrate that allows it to persist in the vertical world of the forest canopy, and in finding these precise microsites, they have diversified into forms as varied as the trees themselves. The resilience of mosses reveals itself most dramatically in their relationship with water, or rather, with the absence of water. When drought descends, when weeks pass without rain and the air becomes parched and unforgiving, mosses do not struggle and die as so many other plants would. Instead, they transform. They dry out, sometimes almost completely, their cells losing the water that fills and animates them, their green fading to brown or gray. To look at them in this state, you might think them dead, spent, finished. But you would be wrong. They have entered a state of suspended animation, a kind of botanical patience, waiting for conditions to improve, for rain to return, for life to resume. The speed with which some mosses can return to active life borders on the miraculous. Within minutes of rehydration, after months or even years of complete dryness, certain species can resume photosynthesis, their cellular machinery awakening from dormancy, chloroplasts beginning once again to capture light and transform it into energy. This is not a slow recovery, not a gradual rebuilding of damaged systems, but rather an immediate resumption of function, as if a switch has been flipped, as if time itself had simply paused and now continues. The moss that was dormant seconds ago is now actively growing, metabolizing, living with the same vigor it possessed before drought struck. During these periods of desiccation, moss metabolism can shut down almost entirely, reducing to the barest whisper of chemical activity. Yet this shutdown causes no damage to the cellular structures that will be needed when water returns. Other organisms would suffer catastrophic damage from such complete drying. Cell membranes would rupture, proteins would denature, the delicate machinery of life would break. But mosses have evolved protective mechanisms, molecular shields that preserve the integrity of their cells even as those cells lose their water. They produce protective sugars and proteins that cushion cellular components, maintaining their structure in the absence of the water that normally provides support and spacing. Temperature extremes that would kill most plants are merely inconveniences to certain mosses. Some species tolerate conditions ranging from near freezing to temperatures that, in experimental settings, approach boiling. They have been frozen solid and thawed, heated until their tissues should by all rights be cooked, and yet they survive, their cells somehow maintaining integrity across temperature ranges that span more than 100 degrees. This extraordinary tolerance speaks to the fundamental toughness of moss biology, to cellular mechanisms that can protect and preserve life under conditions that seem incompatible with it. In deserts, where the sun beats down with relentless intensity and surface temperatures can soar far above air temperature, mosses persist in seemingly impossible circumstances. They endure ultraviolet radiation levels that would damage or kill many other organisms. They survive surface heat that makes the rocks they grow on too hot to touch. During the day, they may shut down completely, dried and dormant, waiting for the relative cool of night or the rare blessing of rain. When conditions improve, even briefly, they activate, growing and reproducing in quick bursts of activity, making the most of every favorable moment. Arctic and alpine mosses face different but equally severe challenges. Here, the growing season is brief, temperatures fluctuate wildly, and conditions can change from benign to hostile in minutes. These mosses have evolved rapid adjustment capabilities, shifting their growth and physiology quickly in response to changing conditions. They can accelerate growth when warmth and light coincide, then slow or stop when cold returns. They adjust their photosynthetic machinery to make the most of the long daylight hours of summer while protecting themselves from the cold darkness of winter. Their flexibility, their ability to respond quickly and appropriately to environmental cues, allows them to thrive where less adaptable plants cannot persist. Because of their resilience, their ability to survive in harsh conditions where other plants struggle, mosses often serve as pioneer species, the first colonizers of bare and inhospitable surfaces. On newly exposed rock, on volcanic ash still warm from eruption, on ground disturbed by fire or landslide, mosses arrive first. They establish themselves in places where soil does not yet exist, where nutrients are scarce, where conditions seem hostile to life. Their presence is not accidental but essential, for they begin the slow process of transforming barren substrate into something that can support more complex plant communities. As mosses grow on bare rock or ash, they begin to break down the surface beneath them. Acids produced by their metabolism etch into stone, beginning the process of weathering that will, over decades and centuries, transform rock into soil. Organic matter from dead moss tissue accumulates in the spaces between living plants, building up the first thin layers of humus. The moss mat stabilizes loose particles, preventing erosion by wind and water, holding material in place long enough for it to begin the transformation into true soil. In this way, mosses create the conditions necessary for other plants to establish, preparing the ground for the succession of species that will eventually follow them. In cold regions, where permafrost lies beneath the surface and the boundary between frozen and unfrozen ground is critical, moss layers play an important role in thermal regulation. The insulating properties of moss mats slow the transfer of heat into the ground during summer, helping to prevent permafrost from thawing. This insulation works in both directions, also reducing heat loss from the soil during winter. The thickness and composition of the moss layer can significantly influence soil temperatures and moisture, affecting not just the permafrost but the entire ecosystem that depends on the thermal regime the moss helps maintain. In forests, particularly in cool, moist climates where moss carpets cover the ground in thick, continuous layers, these green blankets influence the timing of snowmelt in spring. The moss shades the snow that falls upon it, preventing direct solar heating. It also provides insulation, slowing the transfer of warmth from air to snow. Together, these effects mean that snow persists longer over moss-covered areas than over bare ground or areas covered only with leaf litter. This delayed melting affects the timing of water availability, influencing when plants can begin growing in spring, when streams reach their peak flow, when the forest floor transitions from winter dormancy to spring activity. The role of mosses in carbon storage, particularly in peatlands, is difficult to overstate. In these waterlogged environments, one genus of moss dominates above all others, sphagnum, the peat mosses. These remarkable plants have evolved specialized cells that give them extraordinary water-holding capacity. Sphagnum can absorb and hold up to 20 times its own dry weight in water, transforming from a light, crispy material when dry to a heavy, sodden sponge when wet. This water-holding ability comes from specialized hyaline cells, large, empty cells with pores that allow water to enter but resist letting it leave. These hyaline cells are architectural marvels at the microscopic scale. They are dead at maturity, their living contents dissolved away, leaving behind only the cell wall with its precisely engineered pores and openings. Water enters these cells through the pores and is held there by capillary action, the same force that allows a paper towel to soak up a spill. The cells act like countless tiny reservoirs, drawing in moisture from rain, fog, and even humid air, storing it within the body of the moss, creating the saturated conditions that define peatland environments. Sphagnum moss does more than hold water. It actively acidifies its surroundings, releasing hydrogen ions that lower the pH of the water in which it grows. This acidification is not a side effect but an adaptation, for it creates conditions hostile to the bacteria and fungi that would normally decompose dead plant material. In most ecosystems, dead plants decay quickly, their carbon returned to the atmosphere as carbon dioxide within months or years. But in sphagnum-dominated peatlands, decomposition slows to a crawl. The acidic, oxygen-poor, waterlogged conditions that sphagnum creates preserve organic matter, allowing it to accumulate rather than fully decay. This accumulated organic matter is peat, layer upon layer of partially decomposed plant material, much of it the remains of sphagnum itself. In deep peatlands, peat deposits can reach depths of many meters, representing thousands of years of accumulation, thousands of years of carbon locked away rather than released. These peatlands function as massive carbon sinks, storing carbon that would otherwise be in the atmosphere as carbon dioxide, contributing to the greenhouse effect. The role of sphagnum in creating and maintaining these carbon stores makes these humble mosses players in global climate regulation, their growth and spread influencing atmospheric composition at a planetary scale. Beyond carbon storage, moss mats provide other ecological services. On slopes and exposed surfaces, they reduce erosion by protecting soil from the direct impact of rain and the scouring force of runoff. Their dense growth intercepts water, slowing its flow, allowing it to infiltrate into soil rather than racing across the surface, carrying sediment with it. This erosion control is particularly important on steep slopes, on recently disturbed ground, and in areas where other vegetation is sparse. Mosses also trap airborne particles, their dense structure acting as a filter for wind-blown dust and other materials. Nutrients carried on these particles accumulate in the moss mat, and as moss tissue dies and decomposes, these nutrients are released into the soil beneath, enriching it, making it more capable of supporting other plants. In this way, mosses act as nutrient trappers and accumulators, gathering resources from the air and concentrating them where they can benefit the broader ecosystem. Some mosses have evolved partnerships with microscopic organisms that further enhance their ecological role. Certain species host nitrogen-fixing cyanobacteria, microscopic photosynthetic bacteria that can capture nitrogen from the air and convert it into forms that plants can use. Nitrogen is often a limiting nutrient in ecosystems, the element in shortest supply relative to demand, and the ability to capture atmospheric nitrogen and make it available to plants is enormously valuable. Mosses that host these cyanobacteria become nitrogen factories, enriching their surroundings, supporting the growth of neighboring plants, contributing to ecosystem productivity in ways that belie their small size. The presence of moss layers creates distinct microclimates, small-scale environments that differ significantly from the broader conditions around them. Beneath a moss carpet, temperatures are more moderate, extremes of heat and cold dampened by the insulating properties of the moss. Humidity is higher, the moss releasing moisture into the air above it while the dense mat restricts air movement, allowing humidity to build. Light levels are lower, the moss canopy, however thin, casting shade on the substrate beneath. These altered conditions create habitat for organisms that could not survive in the harsher conditions of exposed surfaces, from tiny invertebrates that shelter in the moss to the spores of fungi that need moisture to germinate, to the seeds of plants that require protection during their vulnerable seedling stage. The moss layer becomes not just ground cover but habitat, not just vegetation but architecture, structuring the environment in ways that cascade through the food web and influence the entire community of organisms that share its space. The structure of a moss mat, when examined closely, reveals itself not as a simple covering but as a complex three-dimensional landscape, a miniature world of peaks and valleys, chambers and corridors, surfaces exposed and spaces hidden. Within this tiny terrain, conditions vary dramatically over distances measured in millimeters. Temperature gradients exist between the sun-warmed surface and the cooler depths. Humidity rises as you move deeper into the mat, away from the drying influence of wind and direct light. Light penetrates only so far before being filtered and absorbed by layers of green tissue. These varying conditions create what ecologists call microhabitats, small-scale environments with characteristics distinct from the broader surroundings, and in these microhabitats, an astonishing diversity of life finds refuge. The invertebrate inhabitants of moss mats constitute a hidden fauna, rarely seen but abundant beyond measure. Mites wander through the spaces between moss stems, their 8 legs carrying them on hunting expeditions or grazing missions, depending on species. Springtails, those remarkable jumping arthropods, browse on fungi and decaying plant material, their spring-loaded tails allowing them to leap many times their body length when disturbed. Rotifers, microscopic animals that look like nothing else on Earth with their wheel-like rings of cilia, swim through the water films that coat moss leaves, filtering bacteria and organic particles from the liquid. Nematodes, roundworms of countless species, slide through the water-filled spaces, some feeding on bacteria, others preying on smaller invertebrates, still others parasitizing larger organisms. And then there are the tardigrades, the water bears, those 8-legged microanimals that have captured scientific imagination for their extraordinary resilience, their ability to survive conditions that would kill almost anything else. All of these creatures and more make their homes in moss mats, their populations numbering in the thousands or millions per square meter, an invisible city of life existing in what appears to the casual observer as simple green ground cover. The architecture of the moss itself influences which animals can live within it. Dense, tightly packed cushion mosses create different opportunities and challenges than loosely woven carpet mosses. The size of individual moss plants, the spacing between stems, the depth of the mat, all these factors affect what can live there. Some invertebrates need larger spaces to move through, others prefer tighter quarters. Some require the deeper layers where moisture persists longest, others thrive in the more variable conditions near the surface. The structural diversity of different moss species translates directly into habitat diversity, and this in turn supports a more diverse invertebrate community. A woodland with 10 species of moss will support more kinds of microscopic life than one with only 3 species, not because the additional moss species provide additional food necessarily, but because they provide additional kinds of space, additional variations on the theme of microhabitat. Larger animals also find value in moss. Insects, particularly during dry periods, shelter beneath moss cushions where humidity remains high even when the surrounding air is parched. A beetle or ant finding itself in danger of desiccation can crawl beneath the edge of a moss cushion and enter a world where moisture lingers, where evaporation is slowed by the dense vegetation and the still air trapped within it. For these creatures, moss cushions function as moisture refuges, islands of tolerable conditions in a landscape that has temporarily become inhospitable. The insects may not feed on the moss, may not depend on it for anything other than this shelter, but without it, they might not survive the drought. In the far north, where winters are long and harsh and plant growth is constrained by cold and short growing seasons, large mammals turn to mosses as food. Reindeer and caribou, during times when other forage is scarce, particularly in winter or early spring before new growth appears, feed on moss-rich ground layers. The nutritional value of moss is not high. It does not provide the energy and protein that these animals would prefer, but it is available when little else is, and availability matters enormously when the alternative is starvation. The ability to digest moss, to extract at least some nutrition from these tough, acidic plants, expands the range of conditions under which these large herbivores can survive, allowing them to persist through seasons when more nutritious food is unavailable. Mosses have a peculiar relationship with their own past. Unlike trees that fall and decay, unlike herbs that die back and leave soil enriched but largely unchanged, mosses often grow directly on top of their own dead remains. As the lower portions of moss plants die, their tissue persists, held in place by the living moss above, slowly decomposing but not disappearing. The living moss grows upward, adding new tissue at the top while the old tissue below contributes to an ever-thickening organic layer. In some peatlands, this process has continued for thousands of years, building deposits meters deep, each layer representing a particular moment in time, a horizontal slice through history. The moss essentially builds its own substrate, creating the foundation upon which future generations will grow. This accumulation of organic matter, combined with the moss's effect on water retention and its influence on light reaching the ground, means that moss layers can strongly shape the plant communities that develop above and around them. Soil temperature beneath a thick moss mat differs from that of bare ground. Moisture availability changes. The chemistry of the soil is influenced by the acids and other compounds released by moss. Seeds landing on moss may face different germination conditions than seeds landing on bare soil or leaf litter. Some plant species find moss mats favorable, their seeds germinating readily in the moist, shaded conditions the moss creates. Other species struggle, unable to establish roots through the dense moss into the mineral soil beneath, or finding the moss too acidic, too wet, too poor in nutrients. The composition of the plant community above the moss is, to a significant degree, determined by the moss itself, a case of one layer of vegetation structuring another. The tolerance of mosses for harsh conditions extends even to chemical stresses that would kill most plants. Some species can tolerate heavy metals, growing on mine tailings and contaminated soils where concentrations of copper, lead, zinc, or other toxic elements would be lethal to other vegetation. Others tolerate salinity, colonizing road verges where winter de-icing salt accumulates, establishing themselves in coastal areas where salt spray is constant. These stress-tolerant species are not necessarily thriving in these conditions. They may grow slowly, reproduce rarely, but they survive where others cannot, and in surviving, they begin the process of stabilizing and remediating these disturbed and contaminated sites, paving the way for eventual recovery of more diverse plant communities. The composition of moss communities is not static but shifts in response to changing conditions. Climate change, altering temperature and precipitation patterns, favor some species while disadvantaging others. Air pollution, depositing acids or nutrients or heavy metals, changes the competitive balance among species. Even subtle changes in forest management or land use can ripple through moss communities, altering their species composition and abundance. These shifts can happen quickly, within years or even seasons, as moss species that are no longer well suited to conditions decline while better adapted species increase. This rapid response to environmental change makes mosses valuable as indicators, as early warning systems that can alert us to changes in the environment before they become obvious through changes in trees or other longer-lived, slower-responding organisms. Scientists monitoring moss communities can detect the fingerprints of environmental change written in species presence and absence, in abundance and rarity. A shift toward species that tolerate pollution may indicate increasing air quality problems. An increase in species adapted to drought may signal changing precipitation patterns. The appearance of species characteristic of warmer climates may herald temperature increases. Because mosses are sensitive, because they respond quickly, because they can be surveyed relatively easily, they provide a window into environmental change that might otherwise go unnoticed until it has progressed further and become more difficult to address. The reproductive biology of mosses sets them apart from the flowering plants that dominate most modern landscapes. Mosses produce no flowers, no bright petals to attract pollinators, no sweet nectar or fragrant oils. They offer no fruits, no fleshy rewards to encourage animals to disperse their offspring. Instead, they reproduce through spores, microscopic packages of genetic information launched into the air in numbers so vast that successful colonization of distant sites becomes possible despite the low probability of any individual spore landing in a suitable location. What we see when we look at moss, the green leafy plants that form cushions and carpets, represents the gametophyte stage of the life cycle. Unlike ferns and flowering plants, where the gametophyte is reduced to an insignificant or even microscopic stage, in mosses the gametophyte is dominant, long-lived, photosynthetic, the primary form of the organism. This gametophyte is haploid, meaning its cells contain only one copy of each chromosome, half the genetic complement of the species. This haploid plant produces gametes, egg cells, and sperm cells, through simple cell division rather than through meiosis, for its cells are already haploid. When conditions are right, when water is present to carry sperm from one plant to another, fertilization occurs. An egg cell is fertilized by a sperm cell, creating a diploid zygote with two copies of each chromosome. This zygote develops not into a free-living plant but into a sporophyte, a structure that remains attached to and nutritionally dependent on the female gametophyte. The sporophyte is typically a slender stalk topped by a capsule, unbranched and simple in structure compared to the complex branching seen in the sporophytes of ferns and seed plants. This sporophyte's sole function is to produce and disperse spores. Within the capsule, diploid cells undergo meiosis, reducing their chromosome number back to haploid and creating spores. A single capsule, depending on the species, can produce anywhere from thousands to millions of these spores. Each spore is tiny, often less than the width of a human hair, small enough to be carried by even the gentlest breeze. Their small size gives them extraordinary dispersal potential, allowing them to travel kilometers or even hundreds of kilometers from their parent plant, crossing valleys and mountains, oceans and continents. Most spores, of course, land in unsuitable locations and perish. But with millions being produced, only a tiny success rate is needed for the species to persist and spread. When a spore lands in a suitable location and conditions are favorable, it germinates, but what grows from it does not immediately look like the moss plants we recognize. Instead, the spore produces a protonema, a thread-like structure that spreads across the substrate, branching and growing, sometimes forming a green fuzz or film that is easily overlooked. This protonema is photosynthetic and can persist for weeks or months, gradually building up resources. Eventually, from this thread-like network, buds form that develop into the familiar leafy shoots of the gametophyte, and the moss colony begins to take shape. Not all moss reproduction involves spores and sex. Many species can reproduce asexually, producing specialized structures called gemmae. These gemmae are small clusters of cells, miniature propagules that can be splashed or blown away from the parent plant and, landing elsewhere, grow directly into new gametophytes without the need for fertilization or spore production. This asexual reproduction allows rapid local spread and enables mosses to reproduce even when conditions are not suitable for sexual reproduction, when water is too scarce for sperm to swim, when sporophytes fail to develop. Even more remarkable is the ability of moss fragments to regenerate. Break off a piece of moss, a single shoot or even just a portion of a shoot, and under the right conditions it can develop into a complete new plant. This regenerative capacity means that physical disturbance, rather than eliminating moss, may actually promote its spread. A bird collecting nesting material, an animal brushing past, a falling branch, any of these can break moss into fragments and scatter them, and each fragment is a potential new colony. The dispersal of spores is not left entirely to chance. Most mosses have evolved sophisticated mechanisms to control spore release, ensuring that spores exit the capsule when conditions favor dispersal. Around the opening of the capsule are structures called peristome teeth, specialized cells that respond to humidity. When the air is dry, these teeth bend outward, opening the capsule and allowing spores to be shaken out and caught by the wind. When humidity rises, the teeth bend inward, closing the capsule, protecting the remaining spores from being released when rain or fog would cause them to clump and fall nearby rather than dispersing widely. This humidity-responsive mechanism ensures that spores are released during weather conditions that maximize their dispersal distance. Sphagnum mosses have evolved a different strategy altogether. Their capsules do not simply open and allow spores to drift away. Instead, as the capsule matures and dries, pressure builds within it. The capsule becomes a tiny compressed air gun, and when conditions are right, it explodes, launching its spores several centimeters into the air with remarkable force. This may not sound impressive, but for organisms measured in centimeters, launching spores several times their own height represents a significant achievement, lifting the spores above the boundary layer of still air that clings to surfaces, giving them access to stronger winds that can carry them farther. This explosive dispersal is just one more example of the ingenious solutions mosses have evolved to the challenges of living small in a large world. The mechanics of moss reproduction impose a fundamental constraint that shapes where and when mosses can complete their sexual cycle. Moss sperm are not passively transferred by wind or carried by insects as pollen grains are in flowering plants. Instead, they are equipped with flagella, whip-like tails that propel them through liquid water. These sperm must actually swim, from the male reproductive structure where they are produced to the female structure that houses the egg. This means that fertilization can only occur when water is present on the surface of the moss plants, a film of moisture deep enough for microscopic swimming, shallow though it may be by our standards. Rain provides this water, as does heavy dew or fog, but during dry periods, sexual reproduction simply cannot proceed. The moss plants may be healthy, well-established, producing abundant gametes, but without water to serve as a highway for sperm, fertilization remains impossible. This dependency on water for reproduction has profound implications for moss ecology and evolution. It means that mosses are most likely to complete their sexual cycle during wet seasons, after rainstorms, in climates where moisture is reliably abundant. In dry environments or during drought, mosses may go years without producing sporophytes, relying instead on vegetative growth and asexual reproduction to maintain their populations. The requirement for water also limits the effective distance over which fertilization can occur. Sperm can only swim a few centimeters at most, meaning that male and female reproductive structures must be quite close together for fertilization to succeed. This proximity requirement influences moss colony structure and the evolution of reproductive strategies. Many moss species have evolved a reproductive system where male and female structures occur on entirely separate plants, a condition called dioicy. In these species, some individuals are exclusively male, producing only sperm, while others are exclusively female, producing only eggs. This separation of sexes is remarkably common in mosses, with approximately half of all moss species being unisexual in this way. Dioicy contrasts with the hermaphroditic condition seen in many other mosses, where individual plants produce both male and female structures, allowing self-fertilization or at least reducing the distance sperm must travel to reach eggs. The evolution of separate sexes in mosses remains something of a puzzle, as dioicy comes with significant costs. Male and female plants must grow close together for fertilization to occur, yet this proximity is not guaranteed. Spores landing and establishing new colonies will be a random mix of males and females, and purely by chance, some areas may end up dominated by one sex or the other, reducing reproductive success. Indeed, studies have shown that Diorchis moss species often produce sporophytes less frequently than species where both sexes occur on the same plant. Female plants in some Diorchis species may go years or decades without being fertilized simply because no male plants happen to be growing close enough. Yet dioicy persists and is common, suggesting that it must confer some advantage that outweighs these reproductive costs, perhaps by promoting genetic diversity or by allowing male and female plants to specialize in different strategies. When you look at a large moss patch, a cushion the size of your hand or a carpet covering square meters of ground, you might assume you are looking at a community of many individuals, hundreds or thousands of separate plants. Sometimes this is true, but often it is not. Many moss patches are actually single genetic individuals, clones that have spread vegetatively, each shoot connected to its neighbors through shared ancestry, all derived from a single spore that germinated perhaps decades or centuries ago. These clonal colonies grow outward from their point of origin, adding new shoots at the edges while old shoots in the center may die, but the genetic individual persists, potentially achieving great age and enormous size even though each individual shoot is tiny and short-lived. The timing of moss reproduction is not random but often carefully regulated. Some species reproduce primarily in spring, others in fall. Some wait for particular combinations of temperature and moisture. This timing ensures that sporophytes mature and release spores when conditions are most favorable for dispersal and establishment. A species that times sporophyte development so that capsules mature and dry during the windiest season of the year maximizes the distance its spores will travel. One that times reproduction to ensure spores are released just before the wettest season begins increases the likelihood that arriving spores will encounter the moisture needed for germination. These phenological strategies, the seasonal timing of life cycle events, are as finely tuned in mosses as in any other group of organisms. Some moss species take the timing strategy to an extreme, existing primarily as spores, dormant and waiting in the soil or on surfaces, appearing as visible, growing plants only briefly when conditions become favorable. These ephemeral mosses can germinate, grow, reproduce, and return to dormancy within weeks, completing their entire active life cycle during a narrow window of suitable conditions. The rest of the time, they persist invisibly as spores, waiting with the patience that only dormant life can achieve. In this way, they exploit temporary conditions, appearing after fires, on recently exposed soil, during unusually wet periods in normally dry environments, living fast and disappearing before conditions deteriorate. The physiological mechanisms that control moss growth and reproduction involve many of the same hormones and signaling molecules used by flowering plants, evidence of the deep evolutionary connections between all land plants. Auxins, gibberellins, cytokinins, abscisic acid, these chemical messengers that regulate everything from cell division to stress responses in flowering plants also operate in mosses, often in similar ways. This molecular continuity suggests that the basic toolkit of plant hormones evolved early in land plant history, before the divergence of mosses and vascular plants, and has been retained and modified across hundreds of millions of years. When mosses shift from purely vegetative growth to reproduction, their metabolism must adjust to meet new demands. Developing sporophytes require energy and resources, and female gametophytes bearing sporophytes often increase their photosynthetic rate to supply these needs. The sporophyte itself can photosynthesize to some degree, particularly when young and still green, but it remains primarily dependent on the gametophyte for nutrition. This represents an investment by the maternal plant, energy and nutrients diverted from growth and maintenance into reproduction, an investment that will only pay off if the sporophyte successfully produces and disperses viable spores. Living in intimate contact with moss tissues, visible only under microscopes, are complex communities of bacteria and fungi, microbes that colonize the surfaces of moss leaves and stems, inhabit the spaces between cells, and in some cases even live inside cells. These microbial communities are not merely hitchhikers but active participants in moss biology. Some bacteria help mosses acquire nutrients, breaking down organic compounds into forms the moss can absorb or fixing atmospheric nitrogen into ammonia. Some fungi extend the moss's reach, their filaments exploring spaces the moss cannot access, translocating nutrients back to their host. In return, the moss provides these microbes with sugars from photosynthesis, with shelter, with a stable environment in which to live. The relationship between mosses and nitrogen-fixing microbes is particularly important in nutrient-poor environments. Nitrogen is essential for building proteins and nucleic acids, the fundamental machinery of life, but in many environments it is scarce, locked up in forms that plants cannot use. Certain bacteria and cyanobacteria possess the remarkable ability to capture nitrogen gas from the atmosphere and convert it into ammonia through a process called nitrogen fixation. When these microbes associate with mosses, they transform the moss from a nitrogen beggar into a nitrogen source, allowing it to thrive in habitats where nitrogen scarcity would otherwise limit growth. This partnership has consequences beyond the moss itself, as nitrogen released from decaying moss tissue enriches the surrounding ecosystem, benefiting other organisms. In peatlands, the interactions between mosses and microbes influence processes of global significance. Methane, a potent greenhouse gas, is produced by certain microbes called methanogens that live in the oxygen-poor conditions typical of waterlogged peat. Other microbes, methanotrophs, consume methane, oxidizing it before it can escape to the atmosphere. The balance between methane production and consumption determines how much methane a peatland releases, and mosses, through their effects on oxygen levels, water chemistry, and microbial community composition, influence this balance. The species of moss present, the thickness of the moss layer, the degree of waterlogging, all these moss-related factors help determine whether a peatland is a net source or sink of methane, making mosses indirect players in atmospheric chemistry and climate regulation. Not all microbes associated with mosses are beneficial. Some are pathogens, causing diseases that damage or kill their moss hosts. These moss diseases may seem obscure, of interest only to specialists, but they have attracted attention from plant scientists for a more general reason. Mosses, being relatively simple in structure and easy to grow in laboratories, make excellent model organisms for studying plant immunity, the mechanisms by which plants recognize and respond to pathogens. Research on how mosses defend themselves against bacterial and fungal infections has revealed fundamental principles of plant immune systems that apply across all land plants, from mosses to crops, contributing to our understanding of plant health and disease resistance. The relationship between mosses and neighboring vascular plants is complex and varies with circumstances. Sometimes mosses facilitate the establishment of other plants, their mats retaining moisture, moderating temperature, stabilizing substrate. Seeds germinating in moss may benefit from these improved conditions, and seedlings may find shelter and support as they establish. At other times, mosses compete with other plants, monopolizing space, absorbing nutrients before they can reach roots, creating conditions too wet or too acidic for competitors. Whether mosses help or hinder other plants depends on the species involved, the environmental context, the specific conditions of the site. The interaction can shift over time as conditions change or as the community develops. Unlike plants with roots that draw water and nutrients from deep in the soil, mosses obtain most of their nutritional needs from what falls on them from above. Rain carries dissolved nutrients, washed from the atmosphere or leached from vegetation overhead. Fog condenses on moss surfaces, bringing with it whatever substances it has picked up during its formation and transport. Dust settles on moss, and as water flows over the leaves, these particles dissolve or are broken down, releasing their nutrients. This dependence on atmospheric deposition rather than soil uptake means that mosses are directly exposed to whatever is in the air around them, for better or worse. When the air carries pollutants, heavy metals from industry, pesticides from agriculture, acids from fossil fuel combustion, mosses accumulate these substances in their tissues. They cannot avoid exposure, cannot close stomata or retreat into dormancy when pollution levels rise. The pollutants land on their leaves and are absorbed, building up over time to concentrations that reflect the pollution history of the site. This accumulation, while potentially harmful to the moss, provides scientists with a tool for monitoring air quality. By analyzing the chemical composition of moss tissue, researchers can map patterns of pollution deposition, identify sources, track changes over time. Mosses become natural samplers, biological sensors that integrate pollution exposure over weeks or months, providing information that would be difficult and expensive to obtain through mechanical monitoring equipment. This use of mosses for biomonitoring has become standardized in some regions, with regular surveys analyzing moss chemistry to assess air quality across large areas. The technique is particularly valuable in remote regions where installing and maintaining monitoring equipment is impractical. A researcher can collect moss samples from dozens or hundreds of sites, analyze them in the laboratory, and produce detailed maps of pollution patterns, revealing hotspots, identifying trends, contributing to environmental protection efforts. Mosses have found their way into human-dominated landscapes, colonizing the urban environment with remarkable success. On walls, both ancient stone and modern concrete, mosses establish themselves in cracks and crevices, on horizontal surfaces where moisture collects on shaded north-facing sides. On roofs, particularly older ones with rough surfaces, moss communities develop, sometimes becoming thick enough to hold significant water, to support invertebrate communities, to require removal by homeowners concerned about roof damage. On sidewalks and pavement, in the joints between blocks, in any small depression where water accumulates, mosses appear. These urban mosses are not remnants of former vegetation, clinging to existence in an altered landscape, but true colonizers, species that have found opportunity in the city, that exploit the unique conditions urbanization creates. The presence of mosses in cities softens the hard edges of concrete and asphalt, introduces green into gray landscapes, provides habitat where little else can grow. They absorb water during rain, reducing runoff. They filter dust from the air. They create microclimates that moderate the urban heat island effect, their moist surfaces cooling the air around them through evaporation. In these ways, urban mosses provide ecosystem services, small in scale perhaps but widely distributed, present wherever cities meet moisture, wherever hard surfaces meet the patient persistence of life seeking a foothold. For as long as humans have lived in places where mosses grow, we have found ways to make use of these humble plants. Across centuries and cultures, people have used moss for insulation, stuffing it into the walls of homes to keep out winter cold, packing it around windows and doors to stop drafts. They have gathered it for bedding, creating soft, moisture-absorbent mattresses that were more comfortable than bare straw or rushes. They have used it in clothing and as padding, particularly in cold climates where its insulating properties and ability to wick moisture away from the skin made it valuable for survival. The relationship between humans and moss is an ancient one, born of practical necessity and sustained by the moss's abundance and usefulness. Among all the uses humans have found for moss, one of the most remarkable was the use of sphagnum as wound dressing. This practice has roots stretching back centuries, but it reached its peak during the First World War when conventional medical supplies ran short and battlefield surgeons turned to this natural material out of desperation. What they discovered was that sphagnum worked astonishingly well. Its absorbency meant that a single pad of sphagnum could hold several times more blood and fluid than cotton bandages, and it possessed antiseptic properties that helped prevent infection. The same acidic compounds that slow decomposition in peatlands also inhibit bacterial growth, and wounded soldiers whose injuries were packed with sphagnum often healed better than those treated with conventional materials. The moss was gathered by volunteers, dried, and shipped to field hospitals, where it saved countless lives. Indigenous cultures around the world developed their own relationships with moss, using it in ways that reflected deep knowledge of its properties. Moss served as diapers for infants, absorbent and soft against delicate skin, easily replaced and abundant in the forests where people lived. It was used as towels and as cradle lining, anywhere that absorbency and cushioning were needed. These uses were not mere expedients but reflected a sophisticated understanding of material properties, a knowledge of which mosses worked best for which purposes, how to prepare them, how to dry and store them for later use. Beyond its practical applications, moss has woven itself into human culture in subtler ways. It features prominently in folklore and poetry, often as a symbol of resilience and protection, the plant that covers wounds in the forest, that softens harsh stone, that thrives where other plants cannot. In many traditions, moss-covered places are considered ancient and sacred, the moss itself a sign of age and continuity, a living connection to the deep past. There is something in the appearance of moss, its softness and its greenness, its quiet persistence, that speaks to something in the human imagination. In more recent times, moss has found new appreciation in aesthetic contexts. Moss gardens, particularly in Japanese tradition, are valued for their calm beauty and their evocation of forest floors and mountain slopes. These gardens celebrate the subtle variations in moss species, the different textures and shades of green, the way light falls on moss surfaces, the interplay between moss and stone. Koki-dama, the practice of wrapping plant roots in moss balls rather than potting them in containers, has spread from Japan to garden centers around the world, appealing to people who want to bring living plants into their homes with minimal soil and maximum visual interest. The moss holds moisture, provides structure, and creates an object that is both practical and beautiful. Horticulture has long recognized the value of moss. It is used widely for orchids, which appreciate the moisture retention and the air circulation that moss provides around their roots. It lines hanging baskets, fills terrariums, creates miniature landscapes in glass containers where tiny worlds can exist on a desk or windowsill. In these controlled environments, moss thrives, requiring little care, needing only indirect light and occasional misting to maintain the green vitality that makes it so appealing. As our understanding of environmental challenges has grown, moss is being viewed through new lenses. Moss walls are being tested as air-filtering green infrastructure, installed in urban areas where they can absorb pollutants and particulate matter from the air while adding greenery to concrete landscapes. These living walls require less maintenance than conventional planted walls because moss needs no soil and can survive on atmospheric moisture in humid climates. The potential for using moss in urban design is being explored in cities around the world, from experimental installations on building facades to moss panels in subway stations and shopping centers. Researchers are investigating moss-based materials for applications that our ancestors never imagined. These materials show promise for eco-friendly packaging and insulation, alternatives to petroleum-based foams and plastics. Moss fibers can be processed and formed into materials that insulate, that cushion, that biodegrade completely when their useful life is over. In a world increasingly concerned with sustainability and the environmental costs of our materials, moss offers possibilities that are just beginning to be explored. Some mosses are being studied for their pharmaceutical potential, for bioactive compounds that might have medicinal applications. The same chemicals that allow mosses to survive stress, that protect their cells from freezing or desiccation, that inhibit microbial growth, might have uses in human medicine. This research is in its early stages, but it represents another way that these ancient plants might contribute to human welfare. In laboratories around the world, scientists work with model species like Physcomitrium patens, a moss that has become a standard research organism, helping scientists study plant evolution and stress responses, understand fundamental processes of growth and development, and explore questions that would be difficult to investigate in more complex plants. This tiny moss, growing on agar plates under controlled lights, is contributing to our understanding of how all plants work, how they respond to their environments, how their genes are regulated and expressed. And so we return, at the end of this long consideration, to where we began, to the mosses themselves, growing quietly in their countless habitats around the world. They persist in forests and on tundra, in deserts and in cities, on tree bark and on bare rock, in places wet and places that are dry most of the year. They have been doing this for 450 million years, since before there were forests to grow in, since before there were insects to shelter, since before there were humans to notice them and wonder at their resilience. They will likely continue long after we are gone, these patient green pioneers, these makers of habitat, these accumulators of carbon, these symbols of endurance. In their smallness they contain multitudes, in their simplicity they hide complexity, and in their silence they tell a story as old as life on land itself. The secret world of moss is not really secret at all. It is there, waiting, ready to reveal itself to anyone who takes the time to look closely, to kneel down, to enter that miniature realm where the very old and the very small persist in their ancient ways, asking nothing, giving much, enduring through the long sweep of time with a patience that puts our brief human urgencies into perspective. And perhaps that is the final gift that mosses offer us, not just their practical uses or their ecological services, but this invitation to slow down, to notice, to appreciate the quiet persistence of life in its most modest and most remarkable forms.